Imagine cutting and pasting genes like text on your phone. This is what actually our DNA technology and biotechnology help us do. Hello everyone, welcome back to my channel Genius Minds. In today's video, we are going to learn about biotechnology, principle and process. This video series is going to be best for the CBSE 12th biology students. So let's get started. Biotechnology is, according to the definition, it deals with the techniques that involve usage of the living organism and the product obtained from that organism. So we can make use of the enzymes from these organisms in order to synthesize useful product for the human beings. Now when we talk about the principle of biotechnology, there are many principles related to biotechnology, but the very important ones are, the first one is genetic engineering. So genetic engineering stands for the manipulation of the chemistry of either the DNA or RNA. So you are going to change the chemistry of the DNA or RNA. You are going to synthesize some new molecule and introduce into new organism to change its phenotypic characters, that is physical characters. Very important principle of biotechnology. The second one is maintenance of sterile condition. Sterile means which is free of microorganism, completely pure, so that a particular choice of a microorganism can grow, which is helping out in production of certain important products. Like it can be your antibiotics, it can be your vaccines, it can be the vitamins. So all these are biotechnology derived product where we are synthesizing them from the organism genes. So these are the two very important principle of biotechnology, that is genetic engineering and maintenance of sterile condition, so that the choice of interest of organism can grow and we can synthesize product of our choice. Now moving on to the next thing, when was the first recombinant DNA molecule was synthesized? So since we are using two different DNA molecules from two different organisms, the newly synthesized molecule is known as recombinant DNA molecule. So it was there in the year 1972, two great scientists named as Stanley Cohen and Herbert Boyer. They synthesized first rDNA molecule. What was the experiment all about? So they searched for antibiotic resistance gene from one organism. So if this is the plasmid of that organism and this is the gene which is responsible for antibiotic, so the choice of gene is this one, this area, right? Now what they did was they cut this with the help of some restriction enzymes, the very important molecular biology and rDNA biotechnology. The restriction enzymes are very important. So what they did, they cleaved or they chopped off their choice of gene from that plasmid and they incorporated it into the plasmid of a bacteria called as Salmonella typhimurium. So this choice of gene was incorporated in this Salmonella typhimurium. Now this Salmonella is acting as a vector. It is going to just transport this choice of a gene into the organism. So what organism they have used? They have used an organism over here which is E. coli, Escherichia coli, which shows similarity with Salmonella typhimurium. And it was seen that when this Salmonella typhimurium plasmid carrying the antibiotic resistance gene, the choice of interest, when it was incorporated into E. coli, what the results were really shocking. So now the E. coli too started showing resistance against that particular antibiotic. So that antibiotic resistance gene, what they did, they isolated it from one organism, they incorporated into Salmonella typhimurium where it acted as a vector and then finally it incorporated into E. coli, making it antibiotic resistant. Thus, this molecule, whatever is formed, is known as recombinant DNA molecule, rDNA molecule. So, just an overview if you will see. So, there are the basic three things which are required in rDNA biotechnology or the construction of rDNA molecule. The first one is identification of choice of your interest or the choice of your gene, identification of DNA or gene of your choice. Which gene do you want to select and incorporate into organism? Second one is that introduction into the host organism. In which organism you want to incorporate it. Third one is that maintenance of this choice of gene into that organism. Okay, so that it is successfully transferred to the progeny. So these are the three very important points. So I hope all of you are able to understand. The first one is you should be able to identify the choice of your gene. Second, in which organism you want to incorporate, which vector you are going to use to transfer it from one organism to the another organism. And the third one, it should show its characteristics and it should be able to transfer it to its progeny. So that's all for today's video.